Thank you very much all for being here. My name is Ashfeen. I'm the PhD student in the 37th cycle. I will be presenting my research on the titled Multiscale Investigation of Iodinated Contrast Media Agents in an Engineered Porous Media, which is a Montmorillonite clay. And before I delve too much into my research, I will tell you a brief about my PhD research goal. So I started with this challenge of iodinated contrast media agents in the environment. So some of you might not know what iodinated contrast media agents are. So they are a big family of pharmaceuticals which are widely used in medical industry, mostly for the imaging of X-ray and CT scans. Here you can see an image of a CT scan before and after the injection of contrast media where the brain cancer tumor is clearly visualized. So these kind of pharmaceuticals are used widely in the medical industry, and every year almost 300 metric tons of ICMs are released into the environment. Here is a picture of lots of other pharmaceuticals that has been detected in the surface and tap water in Europe. Now, why are they detected in the tap water and surface water? The problem is that the pharmaceuticals like we study here, iodinated contrast media agents, when they got released into the environment, our conventional wastewater and drinking water treatment is not able to stop their release. As a result, these contrast media get released into the surface waters, drinking waters, and over the years, studies has been conducted where almost every major water body across Europe has been documented to contain a very significant concentration of these contrast media. So here is a plot showing almost microgram concentrations of these pharmaceutical contaminants in Europe. Now, this is where our project came in place, and we were a group of PhDs, researchers, which were tackling this problem to develop engineering and other porous media to help contain, which was trapping, removing, and recycling these contrast media agents from the environment. Now, throughout my PhD, I studied this problem at different scales. So here you can see there is a plot where we start with the scale and the size. So I tried to address this problem at different scales, starting from molecular scale, where I was involved using different types of molecular modeling approaches like density functional theory and molecular dynamics to study how does these pollutants interact with the different engineered porous media surfaces. And on the second scale, I conducted some lab experiments where we developed engineered porous media based on the results we received from molecular studies to build a much more tailored porous media to contain their release. And in the field scale, I conducted some research in the province of Lecco with my industrial partner, Laudereti Holding, where we were able to evaluate different kind of diverse hydrogeological conditions on which these kind of remediation technologies can be applied. Now in this talk, I will be mostly focused on the laboratory scale one. And before I go too much into an experiment, I can give you a brief idea about how does this engineered porous media look like. So on the left, you can see a TEM, which is a microscopic image scan of a clay. Every clay like this one, a colonite Montmorillonite clay, has these layers of these interfoliar layers. Now these layers you can see here, when you look at their structure, these are these two layers which are basically aluminosilicate layers, and the central portion of these layers are filled with these cations. They can be sodium, calcium, magnesium. Our goal here is that we replace these cations with surfactants. Now, what surfactants are basically, they are also cations, but they tend to have these long sort of like a chain associated with their charge. They can be anionic, cationic, non-ionic, or amphiphilic. So in my case, I used this Montmorillonite clay and I modified it with two surfactants. And as you can see, as these surfactants go in this porous media, the interfoliar space of this clay increases. You can clearly see here is the traditional normal clay. And once we fill up with these surfactants, the D space increases. Now there are two reasons, things what we get from the D space. One is the porosity of this clay increases, and second, it also prevents the swelling of the clay and makes it a better porous media to trap in contaminants. Now, to synthesize this kind of engineered porous media, the reaction is pretty straightforward. We start with an unmodified clay. We add in a reaction a surfactant. In my case, I perform a single and double modification. So single modification, I will not go too much details here, but I call it MT1, and with double modification, I call it MT2. Double modification is because we use two kinds of surfactants together. And in the experimental part, my initial goal was to see which of these clay will perform better in context of the removing of contrast media agents. So I started with all these three clays which I have, and first I have to characterize them. So here on this spectrum of the XRD, you can clearly see the first peak which shows us the D space is basically that interfoliar layer, and the size is increased once the clay gets embedded with these surfactants from 14 to almost 18 angstrom. This is FDR spectrum that confirmed the presence of these surfactants on the surface of the clay. Furthermore, we also do the particle size distribution characterization to see how does the clay particle size vary after the modification. And in our case, the MT is the unmodified, the natural occurring clay. And MT2 with the double modification and single modification, the particle size tend to decrease almost to the nanoscale size. Here are some other characterization techniques we involved to help us to better visualize the morphology and chemistry of this clay. The batch experiment, we started with testing all these three clays at different conditions of concentration and time scale. So the experiment was pretty straightforward. We started with a mixture of clay and ICM at a given concentration and adsorbent dose. We used a mechanical shaker to check the removal capacity, and then the samples were analyzed in a UV visible spectrum. Now, of course, we had three different clays to test, and once we tested all these three different clays, here are the results. And you can clearly see the double modified clay showed almost 94% removal rate at the lowest concentration of 10 milligram per liter. Now, since this clay had a better porosity, had a better a higher surface area, it ended up having the best performance for the removal of our contaminant. And the unmodified naturally occurring clay was almost showing negligible, almost 20% removal rate. So we ended up studying our MT2, which was our dual modified clay from now on. We checked it at different concentrations. As the concentration increased, the adsorption capacity and the removal rate decreases. We also tested it at different pH. So here you can see once the batch experiment was conducted at more acidic pH like 5, the adsorption rate and the capacity tends to increase, which is basically showing that since the acid tends to activate this clay more. But at the very highly acidic and a very highly alkaline pH, which is not something that we really find in the environment, the adsorption capacity and the rate decreases. But overall, we had a very significant removal performance at the neutral 7 pH. So after our batch experiment was done, we tried to model both the kinetics and the adsorption isotherm associated with this adsorption process. To model the isotherm and the kinetics, we used a variety of conventionally utilized adsorption isotherm model like Langmuir, Findlay, Hemken, and Sips model. And for the kinetics of the batch, we used pseudo first order, pseudo second order, and interparticle diffusion models. Now, each of these models have the parameters that needs to be calibrated in order to fit these models. And in our case, we used the maximum likelihood framework to optimize, to calibrate the models and to optimize the model parameters. And furthermore, we used different model selection criteria. In our case, we used the ICMs, Bayesian, and Kashup information coefficient. In the case of this methodology, we tend to put more focus on the KIC because it takes in account the determinant of the covariance matrix, which gives us a better idea about the uncertainty of the parameters as well. Now we use the Bayesian methodology with the posterior probabilities in case of the model selection criteria here. And just to give you an idea, here are the results from the batch experiment of the isotherm. Now, on the top of the table is the optimized and model parameters and their corresponding standard deviation. And here is the posterior probabilities associated with each metric of ICAN, Bayesian, and KIC. So here we can see, although the R-square determination coefficient of Langmuir and SIPS were almost same, but the KIC posterior probability of Langmuir is significantly higher than SIPS model. So in our case, what we did, we end up selecting both of these models for the mechanistic explanation of this adsorption process. So two of the parameters that are very important to explain here are the QM, which is the total capacity of the adsorbent, which is about 61.5 milligram per gram. And the current industrial standard, like activated carbon, has almost capacity equivalent to our clay. And for the kinetics of our adsorption process, since we had variety of experiments conducted at different concentrations, you can clearly see that the interparticle diffusion model was not selected based upon the KIC posterior probability, but we had the combination of pseudo first order and pseudo second order at some concentrations, but mostly it was the pseudo first order that end up being selected based upon the KIC posterior. Now, these are the two parameters which we calculated at different concentrations of the experiments. Here you can see the capacity parameter tends to increase, but reaches at a saturation at very higher concentrations, which is very common in these types of clays. Now, once we evaluated the batch experiment to check the performance of the clay, but in a real world applications where we try to utilize these kind of engineered porous media in a drinking water treatment applications, we must perform some kind of continuous column experiments. So in our column experiment, we started with this type of a lab scale column with the dimensions of about 60 millimeters by 92 millimeters. And the column consisted of multiple layer settings. So first of all, before going into two details about the column properties, I will tell us about more about the experimental setup. So we had a injection pump. We used this instead of peristaltic pump because it has a much more controlled flow rate and a continuous flow rate. And in our column adsorption setup, we used an upward flow setup, which helps us to prevent the channeling during the injection of the contaminant. Also, we used an effluent sample collection, and the characterization of the concentration of the outlet was almost the same as batch experiment, where we used the UV spectrum. And we measured all these concentrations on the outlet, which helps us to plot the breakthrough curve as a function of pore volume or time. Now, about the column setup, since these clays, once they come in contact with water, they tend to swell. So to address this challenge, we had these kind of multi-layer setup where we had this composite sand plus clay layer, which is sandwiched between these two sand layers. And the middle layer is basically a mixture of the sand with our engineered clay porous media. And we did this in three separate columns where we added the sand with different ratios of our engineered clay. And we end up having three separate columns, each with a different percentage of the engineered clay added to the central layer. And we used the water saturation experiment to calculate the pore volume and the porosity. And you can see as the clay is increased in the middle layer, the porosity tends to decrease, and so does the pore volume. And after that, here is a simple representation of one of the tracers, which was injected in the sand-only column to check other parameters like the dispersivity and the hydraulic conductivity. And after these tracers was injected, we had the water. First, we inject the pump with just water to have the fully saturation, removing all the air bubbles. And once the column was saturated with water, we injected our contaminant. Now that was injected at different flow rates, at different concentrations, and also in all these three separate columns. Now you can see clearly here that there's a gradient of this methylene blue tracer forming here. Of course, in the lower flow rate, we tend to have more sample intervals. So of all the experiments we conduct, I will show you the 4 extreme cases. One is the standard, the base experiment, where we have the lowest flow rate, lowest concentration, and highest adsorbent mass. The second one is where the flow rate is the highest. Third one is where the concentration is highest. And fourth one, where we have almost just 0.5% of adsorbent in the system. And in the breakthrough, you can clearly see the standard experiment had a later saturation and a later breakthrough. And this is plotted as pore volumes here. And as the concentration increases, we have the earlier breakthrough, earlier saturation, and so does when we have higher flow rate. And when we have almost no adsorbent, less adsorbent in the system, we have almost a plug flow. Now, I conducted all these experiments. Here you can see the experiment was conducted from experiment number A to I. And how we did it, we have this standard experiment at a adsorbent clay fraction of 5.3%, the flow rate of 0.4 ml per minute, and an initial concentration of 100 milligram per liter. And we end up increasing the flow rate, increasing the concentration, and decreasing the clay fractions. And these are some of the common metrics that we calculated, something like mass transfer zone. The saturation time is basically when the outlet concentration reaches almost 95% of the inlet. And you can see clearly as the clay fraction increases, we had a higher saturation time, which is quite normal. And so does when the flow rate increases, the saturation time will decrease. And when the concentration increases, the saturation time also tends to decrease. Here, the adsorption parameter, adsorption capacity, which is the QE, is significantly lesser than our BAT. Now, since this is a column setting and there is less contact time, as a result, our adsorption capacity decreases from 50 to 11 microgram per liter. And as the concentration increases, we have this kind of saturation happening where we can no longer adsorb more contaminant. Now, much like our batch system, we also modeled our column with different kinds of numerical models, something from which are conventionally used in adsorption breakthrough curve modeling. We use the Thomas, Young Nelson, Adam Bohrut, Clark, and Yon model. Now, all these models are formulated based upon some assumptions and simplified some certain consideration based upon the conventional adsorption, dispersion, and advection models. And some of these models, for instance, if I go into the Thomas model, it takes into account the Langmuir isotherm behavior of the adsorption process, and the 2 parameters that are supposed to be optimized or calibrated here is the QTH and KTH, which is basically the rate and isotherm parameter. So each of these models were calibrated much like our batch isotherm. We used the same maximum likelihood model calibration and our Bayesian model optimization strategy here. So these are all these models fitted to these different kinds of experiments. And here I want to show you the experiment A and E are the experiments that are conducted in basically the lowest flow rate and the highest flow rate and the lowest adsorbent. And you can see there is almost like an instant breakthrough. So once we run our model and we calculated the posterior probabilities, you can see almost the determination coefficient is almost same. But using the KIC model weights, we can see for these extreme concentration experiments like experiment A and experiment C, we have the Thomas model, which are quite simplified representations of the model, where for other experiments where we had the later breakthrough, we have the Yan model, which was selected based upon the KIC posterior. And these are the 2 Yan model parameters. I will not go too much detail about explaining every parameter and what does it explain mechanistically, but if you want to go too much details, you can let me know. So overall, this model was also modeled, this experiment was also modeled using the advection dispersion. And we also used some molecular dynamics modeling to also model the molecular level, this experiment at the molecular level. And some of the key conclusions were like we were able to achieve 93 to 94% removal rate of our engineered media with almost 61.1 milligram adsorption capacity, which is quite better than activated carbon in this scenario. And we also used our continuous flow experiment where we successfully tested it for real world applications. And that's it. I would also thank to Horizon Europe for funding this research as a part of the Marie Curie Remedy project. And this is the link to our website if you want to know more about our project and other research on this topic. Thank you so much.
Thank you. Thank you, Ashfeen, once more for the presentation. Now we will get questions from the audience. Anyone who has any curiosity on what was presented? Okay.
Thank you very much for the presentation. It was really full of results. So I see the workload that you had with these experiments. I have two curiosities. One is if you plan to use the batch isotherms and kinetics results to predict the breakthrough and say if the breakthrough results you had in lab are sort of matching with the models. Yeah, maybe you can.
I think we can take those as an initial guess if we want to model our breakthrough curve. But then if you take, let's just say, Langmuir model parameters,